I'd like to welcome all of you to today's CAP Today webinar entitled Molecular QC, Enabling Next-Gen Sequencing and Precision Medicine in the Clinical Laboratory. I'm Bob McGonigal. I'm the publisher of CAP Today, and I'll be the moderator and host today for this very exciting program. You're all making history today as participants. This is the largest web audience that we've ever had for this continuing series of commercial webcasts, and I want to welcome you one and all. This is one of now a series of commercial webcasts that we've been putting on over the course of the last 2 or 3 years. I think it's very important that you understand that the CAP Today does not endorse any of products or services that may be named herein. And any of my own comments are completely personal. They're not to be construed or taken as official comments for either the CAP Today or much less the College of American Pathologists. They're all my own. It's important to understand that this is a commercial webinar and is made possible by a special education grant from Horizon's Diagnostics Division. On the issue of housekeeping, before I introduce our first speakers, I want to let you know that you'll be hearing this predominantly through your computer speakers. You can call a phone number to access, but it's principally over the speakers that you'll be hearing. And if you wish to make a comment or ask a question, you should feel free to do so at any time. You'll see a box labeled Q&A. It's right underneath the overheads, and you can type in there, and we will, at the conclusion of all the formal presentations, answer questions, field comments, and generally engage with all of you as best we can. And now I'd like to briefly introduce our panel. Our first speaker will be Dr. Jonathan Frampton from Horizon Diagnostics. He'll be followed by Josh Deigan from UCLA, and our final presenter today will be Dr. Chris Corless from the Knight Diagnostics Laboratory at Oregon Health Science. I'll introduce each of the speakers more formally when it's their turn to go. But first, I've asked Dr. Jonathan Frampton of Horizon Diagnostics to lead off with a brief description of where we are in the state of the art in next-gen sequencing, and to set the scene for us, if you will. So please go ahead, Dr. Frampton.
Cool. Thank you, Bob, and thank you, everyone, for joining us today for the show. You'll all be very pleased to know it's nice and sunny in Cambridge, so we've got a sunny experience coming up. I'd like to start by thanking both Dr. Deigan and Dr. Corless for making this possible and joining us for the webinar. I'm really looking forward to their 2 talks that are coming up. So I'm Jonathan Frampton, and I'm the Associate Director of the Diagnostics Division at Horizon Discovery. And what we do at Horizon Discovery is we are passionate about delivering and developing high-quality reference standards for use as independent external controls. And what I'd like to touch on today is very much how they can be used, or how, as you are starting to adopt next-generation sequencing into the clinical workflow, how could you actually use reference materials or reference standards to ensure that particular validations are working, and you know the quality and the confidence of the data you're getting out of your workflow. And a question I always like to challenge the audience when I'm presenting is this one: what is the impact of assay failure in your laboratory, and how do you monitor for it? It's an important question to ask when you're running routine assays, and it becomes even more critical when you're looking to incorporate new technology within the laboratory, because whilst new technology always brings great advantages, and I'll touch on them in the next slide, there's also always sources of variability that can creep in when you are adopting new technology. And I'm going to flag up this question a few times, and I would challenge that all of you in the audience, you do consider this as we go through the webinar today, to really understand, okay, what would happen if the assay gave you a false positive or a false negative, and actually what steps could you take to mitigate against that? So one of the biggest advantages of next generation sequencing, and there are others, but I'm going to focus on this for today, is you can take your tumor sample, you can extract the DNA from that, and then put it through your next generation sequencing workflow. And then what that allows you to do is from one sample, you can then test the status of each gene, and that could be a panel of 100, it could be a panel of 200, and it's so much more cost effective and gives you a greater speed to get the result versus having to take one DNA sample, run that for say a KRAS Sanger sequencing at one loci, like the 12 and 13 together versus the 659, 61 together versus the 117 or the 146. So instead of doing multiple individual reactions, you can just put one sample in your workflow and you get the mutation status across all of these genes outlined here. Now that's for me one of the biggest plus points of next generation sequencing. And then what's kind of the thing to consider as you're adopting it into the lab is where can variability actually creep into this process? And so what I've put together here is just a simple schematic of the next generation sequencing workflow, where you start with your tumor sample, you go through DNA extraction, you quantify it, you do a library prep, you sequence, goes through some bioinformatics steps, you can then analyze and take action. Now, throughout this workflow, there's always going to be variability creeping in, and it's the same whether or not you're doing next generation sequencing or you're doing Sanger sequencing or qPCR. The important part is understanding where that variability is. And so, for example, with DNA extraction, there's different kits. Some kits are optimized to give you high-quality DNA but may give you less yield. Some kits are optimized to give you very high concentration samples, but then you may get lower quality DNA, so you may get a mixture of high molecular weight and low molecular weight DNA. There's always an equilibrium where you're balancing the concentration, the yield, and the quality. When you're looking at the DNA quantification, this is a hot topic because depending on whether or not you go down fluorometric measurement or a spectrophotometer, it could give you a different answer, and it could either over or underrepresent the amount of DNA you actually have in the tube. And if you were to overestimate how much DNA you actually have, what would happen is you would then underload DNA into your library preparation step, which would then have a cascade of problems when you then get to the analytical part of your workflow. And so what we're looking to do and passionate about doing is developing a panel of reference materials where you can start to answer some of the questions of, okay, what's the best kit for me for doing the DNA extraction? What's the best method for DNA quantification for our lab? Is there any library preparation bias? Is there any bias in the sequencing? It's important that I do point out that all of our reference standards and reference tools that we provide are currently for research use only and need to be used appropriately in that space. And on that note, one very attractive reference tool we have developed, I believe Josh will touch on in a few moments, is we have genome in a bottle reference material as FFPE samples, and so you can actually take a genome in a bottle, a well-characterized genome, all the way through this whole process, and actually see, okay, how good is the DNA extraction, how good is quantification, and then how does that impact through library prep and bioinformatics. That brings me nicely back around to the question I posed at the start, and like I said, I really would challenge you to think about this as Josh and Dr. Deigan and Dr. Corless go through their talks. What is the impact of assay failure in your laboratory, and how do you monitor it, in particular when you're looking to adopt next-generation sequencing into your laboratory. I'm really looking forward to the next couple of talks, and I thank you very much for listening to my brief introduction. And I will join you at the end for summing up and enjoy the questions and answers. Bob, over to you.
Dr. Frampton, thank you very much. You've done a very nice job setting the scene for us and raising, I suppose, the fundamental question that are on the minds of everyone who contemplates using next-gen sequencing in the clinical laboratory. Dr. Frampton will be remaining with us, and if you have questions or comments for him, it's very easy to do so. Simply type into the Q&A box. You can do that at any time during this webcast. I will, at the end, go through and triage and collate questions and comments. Also, I want to let you know that this webcast will be reproduced in its entirety, both the overheads and the audio, within about a week or 10 days following this program, and you can look at it on captodayonline.com, which is our Cap Today magazine's website. And now it's my great pleasure to introduce to you Dr. Josh Deigan of UCLA. He's the Associate Director of the UCLA Molecular Diagnostics Laboratories, and he's Assistant Professor of Pathology and Laboratory Medicine at the David Geffen School of Medicine at UCLA in California. He's also a Fellow of the American College of Medical Genetics and Genomics. They are getting two words out of one G, so I applaud them for their efficiency. And he's going to speak to us on validation and QC challenges for germline panels, exomes, and genomes. Dr. Deigan.
Thank you very much, Bob. And I'd also like to thank, of course, Cap Today and Horizon Diagnostics for supporting this webinar, as well as Drs. Frampton and Corless, who are presenting with me. So as Bob mentioned, I'm going to be talking to you today about some validation quality control challenges, specifically focusing on the germline setting, panels, exomes, and genomes. But a lot of what I'm going to talk about will also be applicable in the somatic context as well, which Dr. Corless is going to focus predominantly on. So I have no disclosures. And the method that we're really focusing on today is next-generation sequencing, which has certainly exploded in the clinical setting over the last couple of years. And really, it's important because what we're now able to do is to sequence many more fragments than we ever used to in a massively parallel fashion using a variety of different chemistries, platforms, sequencing abilities. But at the end of the day, we're trying to determine what the sequence of a particular fragment is and then maybe how it aligns to a particular region in a patient's genome. So really the question that a lot of us I know are asking for those who are either doing it already or want to do it is, how do I provide clinical next-gen sequencing in my laboratory? So the outline for my talk today is going to be focused on three areas. I'm going to start off talking about clinical next-gen test development, then move on to some germline next-generation sequencing test validation challenges, and then end with a couple of examples of some germline next-gen test quality control challenges. So thankfully, for those who like literature references, there are a number of good references that exist. I was part of the work group that wrote the clinical lab standards for next-gen sequencing from the American College of Medical Genetics and Genomics. And it's good to know that these are published, but that they do focus mainly on germline testing. And I would also point people's attention to these two other very good reference articles, one from the Association for Molecular Pathology and the other from the CDC, both touching on some of the validation quality control challenges that I'm going to talk about today, mostly in the germline setting as well. So let's start with test development. So test development is really where you set the stage for everything else that you're going to do. And what's important to decide is what type of test am I going to offer. So for germline stuff, basically there are three types. A gene panel, which in the germline setting typically involves sequencing of full coding regions, which in order to get full coding region sequencing, there may be some Sanger sequencing fill-in involved. There's also exome sequencing, which really is just a very large panel where you attempt to sequence the coding regions of all genes. And we typically get, in the past, it was maybe closer to 95% disease gene coverage, but now with some improved chemistries, we're getting closer to 98 or maybe 99. And then finally, there's genome sequencing, where you attempt to sequence the entirety of the genome. Most of the laboratories offering germline clinical next-gen testing today would be offering gene panels or exome sequencing. And so basically, if you think about having a next-gen platform, which I've depicted in the middle as just having a fixed sequencing capacity, if you were to run a smaller panel, such as either a small gene panel or maybe even this would be applicable to a next-gen hotspot panel, you would have only a couple of fragments or a few fragments. And so each of those fragments could get sequenced a certain number of times. But using that same platform, should you choose to load an exome or a genome or a multiplex, maybe a set of panel samples, now you have more fragments that you're requiring sequencing, and so each fragment can then be sequenced a fewer number of times. So you just have to keep this in mind. And so naturally there are different instruments that have evolved into the marketplace, some of which are shown here. The one on the top left is certainly good for genomes, exomes, because it has a high sequencing capacity. And the ones on the bottom right are better for maybe gene or variant panels because they have a lower sequencing capacity. But every laboratory needs to decide for themselves which instrument is most appropriate for the type of test they want to offer. And so from a regulatory perspective, it's good to know that according to the FDA, these are really all research instruments except for a couple of them. So what we're talking about when we're bringing up one of these next-generation sequencing tests is the validation, not the requirement for a verification. So moving on to test validation. So test validation is really where we try to understand how our test is going to perform when we launch it in the clinical setting and use it on patient samples. So there are various parameters which we try to assess in order to understand how it works, some of which are shown in this diagram here. And there are also very nice reference articles, this one published by the College of American Pathologists a number of years ago, which talk about some of the parameters which laboratories need to address in order to appropriately validate or verify a test. So today I'm going to focus on the one highlighted in red here, which is accuracy. Accuracy and how one approaches accuracy for a next-generation sequencing test will really help set the stage for how one chooses to address all of the other relevant parameters which a laboratory will need to do. So that's why I'm going to focus in on accuracy for today's talk. So one approach that somebody might take to validating the accuracy of a next-gen test is, well, let's compare our next-generation sequencing results to those from another clinical laboratory performing the same assay or the same test. And we're calling that as a laboratory the gold standard. And so typically this is how one might choose to go about this. So in the setting of a single gene germline test or maybe a small variant panel like a CF variant panel, we would typically try to acquire multiple positive and negative samples, try to get each positive sample to have at least one clinically relevant variant. But the question then is, so what if you're actually looking at all of the genes, such as exome sequencing, or maybe a large number of genes, like a large gene panel? How can you approach this then? Well, it's good to remind everybody that actually when we're dealing with something like an exome sequencing test, which we have actually offered on a clinical basis since early 2012, when you look at the number of variants in an individual sample, we're dealing with, as this diagram illustrates, on the order of 15, maybe to 20,000 variants. And that's not excluding synonymous ones. That's not looking at ones that may be more prevalent in certain populations than others. So that's actually a lot of information that you can utilize for test validation. So I like to kind of call this, is one validation sample enough? And when you think about that question, it sounds funny to say, and yet when you think about validating a next-gen test, it makes a lot of sense. And that's just because it is possible to compare many variants from a single sample. However, I would say that we nor any laboratory should just validate a next-gen test based on one sample, and that's based on various differences that exist between two samples, such as matrix variability, different genomic alterations, and I'm sure others can think of other sources of variability that may exist. So that being said, if one wanted to use all of that variation in a single sample, can you actually compare all the variance in a single sample between two labs? And really the question you're asking is anyone actually performing the same tests that you're performing? And the answer is sort of yes and no. And so this is some unpublished work that was presented at a meeting a couple of years ago, and there are other pieces of work like this. But this is just showing a single genomic DNA sample that was sent to 5 clinical laboratories, all of which were performing clinical exome and genome sequencing. And really all we asked them to do was run their test, use their bioinformatic pipeline, and then report back all of the variants that they see. And we chose to focus on just the variants that were present in the exome for this because that was the region of overlap between all of the labs that participated. And what you can see is that out of the 20,000 or so variants that were detected, there was roughly 90% agreement between all 5 labs, and this diagram just shows the single nucleotide variants. And so there are all these other overlaps between the labs. Obviously, some labs have a number of variants that only they saw and nobody else did. There are variants that 4 out of 5 labs saw or 3 out of 5. But it's just good to know that you just naturally get this source of variability when you're trying to compare variants from a single sample between 2 or multiple laboratories. So I would certainly argue probably not everybody is performing the same test that you are, even if the concept is the same, like 2 labs performing exome sequencing. So it is challenging to compare next-gen data, and I would just say that any laboratories who are wishing to validate their next-gen test using this method take this into consideration. So another approach to validating the accuracy of a next-gen test is, well, if we're not going to compare to another lab, how about let's compare our next-generation sequencing results to those from a gold standard method? And so I think in molecular diagnostic testing, Sanger sequencing is traditionally considered to be the gold standard, which leads us to the question, is Sanger sequencing an appropriate gold standard? So this is just a quick overview of a recent clinical scenario here at UCLA where we had a female through our clinical genetics practice, and you can see there was a number of clinical indications, a definitive suspected diagnosis, glycine encephalopathy, which is a autosomal recessive condition, and so a number of genetic tests were ordered, both Sanger sequencing and targeted exonic microarray, all of which came up negative, which led people to naturally scratch their heads. So exome sequencing was ordered next, and ironically enough, what we found was a homozygous variant in this GLDC gene, which was one of the genes that came up negative for a Sanger test and a deletion duplication test. And so I'm just pointing this out because we've had a number of these scenarios where a prior Sanger sequencing test or gene panel was negative and exome sequencing was positive for one of the genes that made a lot of clinical sense. So how is this possible? And why is Sanger sequencing no longer the gold standard? Well, the usual thing that people point to is this potential for allele dropout due to polymorphic positions underneath primer sites. So in the figure in the middle that you can see, we have 2 copies of a particular variant. The one on the top would be the reference sequence. The one on the bottom would be the alternate sequence. And so if the red star happens to designate a polymorphic position, which disrupts primer binding, this individual may only have their reference allele amplified, thereby masking this alternate allele, so this person would be incorrectly classified as being negative for this position. Also, when you think about next-generation sequencing and coverage, so typically we might try to get on the order of 30, 40, 50, or maybe hundreds of independent reads for coverage for an exome, or maybe thousands of independent reads for a panel. For Sanger sequencing, I like to think of it as having really only 4 independent reads. and that's because you really only have 2 primers that can bind to both copies of the chromosome. So if you're comparing a method with 4 independent reads versus another one with maybe 50 or 100 or 1,000, it's pretty easy to see the difference. Also in the mosaic context for the germline setting, but also applicable in the somatic context, would be the limit of detection between Sanger sequencing and next-generation sequencing. So what I'm showing here is just a generic overview of 5 cells, 2 of which have an alternate allele in heterozygous form, and the other 3 are complete reference. So this may designate a situation where you have a 40% mosaic content, namely 2 out of the 5 cells have this alternate allele, and so in totality there's a 20% allele frequency here, which most likely a Sanger sequencing or next-gen test would pick this up. However, in this scenario, where maybe this is a 20% mosaic content or a 20% somatic content, now we have a heterozygous allele in this cell, 10% allele frequency. Sanger sequencing may not pick this up, but next-generation sequencing likely would. So the conclusion really from these couple scenarios is that there actually may not be a different gold standard method to compare next-generation sequencing to anymore. And I would argue that next-generation sequencing is the new gold standard method. And one thing that I would point out at the bottom is that for comparison for an exome or a genome level test, some laboratories may use SNP arrays for comparison to get at an increased number of variants, but I would just caution those laboratories on looking at the overlap of the SNP array between their regions of interest just to see if there are enough variants that they want to use for comparison between their next-gen test and the array results. So the third and final approach which I'll propose for validating the accuracy of a next-gen test is if we can't easily compare results between 2 labs and there isn't another method that seems like it's the gold standard, then how about we compare our next-generation sequencing results to the results from a well-characterized reference material, which is presumed to hold the truth for those variant positions. So then the question is really, where do I find one? Well, thankfully, as Dr. Franklin alluded to earlier, there's a group and a number of people that have been working on this for quite some time. And so I'm just showing a recent publication from this journal where the group and really the consortium who designates themselves as Genome in a Bottle described their process. And so this slide was borrowed with permission from Genome in a Bottle. But what this highlights is that what this group is attempting to do is to develop well-characterized reference materials that will be available for purchase for clinical laboratories wishing to use them. And so this started out from the Genetic Testing Reference Material Project, and there were cell lines available through Corless. And actually within another couple weeks, there will be the first well-characterized reference material from Genome in a Bottle available through NIST. And so I would encourage everybody to view their website, contact them if you have any questions, because I think this will be an exciting area of development for clinical laboratories wishing to use these types of reference materials. And the last comment about reference materials that I'll point out is that laboratories recently received the first formal proficiency testing for next-generation sequencing, which I'm showing a snapshot here, where laboratories will be asked to essentially look at specific variant positions. This one happens to be a position in MLH1, and report back whether they see a variant, its zygosity, what type of variant it is, so on and so forth. And this is using a different genome than the Genome in a Bottle project. So these will be very interesting results to see once they're available. So finally, I'm just going to talk a little bit about some quality control challenges that we have seen in the germline side. So when you're thinking about quality control on the germline side, really the question you're asking is, should you run something as simple as a positive control and a negative control or a no template control with every exome run or every genome run or every panel run? And really, with something like next-generation sequencing, the question is, could you afford this, and does this make sense? And moreover, how would you even define a positive exome or a positive genome, knowing that there are so many variants that exist in all of the different regions? So thankfully, we have kind of a history of this already with Sanger sequencing, where typically Sanger sequencing tests don't require a positive control, because as long as all of the peaks and the sequence that they produce match the sequence of the gene which you're attempting to sequence, that is really its own positive control. And naturally, you would, in that setting, run a no-template control, but we don't require a specific variant control for every Sanger sequencing test that we do. So why can't next-gen data function as its own quality control as well? So I like to think about this as, if you think about treating genomic DNA as its own next-gen sample type, which is actually something that a lot of clinical laboratories are now subscribing to me it makes a lot more sense. And so I would certainly say that laboratories should validate your next-gen panel exome genome using your own routine, either automated or manual DNA extraction method. But sometimes, for certain clinical scenarios, other institutions may want to send you genomic DNA from their clinical laboratory instead of a new blood sample or something to that effect. And so really then the question is, do you need to validate all DNA extraction methods that could possibly exist? Well, I don't think that's possible. So one approach is, let's have some defined quality control criteria which confirm that your next-gen sequencing was acceptable. So the way I think about this is, what should a normal human exome or a genome look like? Well, there are a number of parameters that you can see listed here, which after you sequence enough panels, exomes, and genomes, you kind of start to get a feel for what normal variation looks like. And so on average, there is a certain range in which every exome, for example, has as far as the total number of single nucleotide variants that we would detect. Also the number of insertions, deletions. And we can also look at things like transition transversion ratio, ratio of heterozygous to homozygous variants. And so for those who like to plot things out using Levy-Jennings plots like I have shown at the bottom, you can attempt to catch any outliers using whatever your lab decides as the range which they want to use, whether it's plus or minus 2 standard deviations, plus or minus 3, so on and so forth, and these things become very valuable quality control metrics or quality indicators. Another idea that has predominated the germline next-gen sequencing world is this concept of confirmation through typically Sanger sequencing for all reported variants. So we, in thinking about this, don't think that this inherently makes a lot of sense. And I would argue that for laboratories performing a gel electrophoresis test or maybe a real-time PCR test, is it routine to confirm all positive findings by Sanger sequencing for either of those methods or another method that another lab might come up with? And I think for the most part the answer is no. So what we essentially did is, for the first year and a half of offering clinical exome sequencing, we confirmed all reported variants, both single nucleotide variants and insertions and deletions. After that time, we essentially validated our ability to do away with Sanger confirmation for high-quality single nucleotide variants and published it in this paper in Genetics and Medicine last year. And so currently, we still do Sanger confirmation for all low-quality SNVs and all insertions and deletions. Any variants that we don't confirm, we evaluate those reads manually. And just to know, so I showed a quality score cutoff of 500, which is for our lab only and is not generalizable to other laboratories. So other laboratories wishing to do the same thing would need to do their own investigation. I will say that this is how we approach our germline exome tests, and that for our somatic next-gen panels, which we currently offer both for colorectal and lung tumors, we don't currently do Sanger confirmation for positive variants that we find there, as I say here, mainly due to the difference in sensitivity of methods. And then for the CAP checklist, really the CAP checklist leaves it up to the individual lab directors to decide whether or not to confirm. So I would just encourage everybody to decide for themselves using a well-thought-out, data-driven process how they want to approach this. And then one last point which I'll mention, just come back to this Sanger sequencing gold standard approach. So from a quality control perspective, sample mix-ups can be easily missed through a typical Sanger sequencing test for a lot of the reasons I gave before. And so a lot of people like to use that as an argument for why Sanger sequencing of a next-gen variant is still important. And I would argue that in the context, again, for exome sequencing, where our preferred option is what we call trio exome sequencing, where we're sequencing both the father, the mother, and the proband, you can actually fairly easily spot any sample mix-ups because of the number of variants that are expected to be shared with each parent. And so here you can see roughly half from the father, half from the mother, and yet in this scenario, should we ever encounter a sample where there is no good match to the father, there maybe is no good match to the mother, this would easily be picked up as a sample mix-up using a next-gen sequencing approach. And just finally, I will say that even when all else in the lab fails and all of the relevant quality control indicators and parameters might miss something, we thankfully sit around on a weekly basis and talk about a lot of our next-gen sequencing cases, specifically all of our exome cases. And so it's very enlightening to hear things that come up during the discussion, and I would just postulate that colleagues can certainly be a good source of quality control and can catch things that maybe the lab is not set up to detect. So in conclusion, I will say that next-generation sequencing is, in my mind, the new gold standard method for clinical molecular diagnostic testing, and that although next-gen validation and quality control is challenging and you may have to utilize slightly different approaches, all of the principles that it's all based on are definitely the same that labs are comfortable with. And so with that, I'm going to turn it back over to Bob.
Dr. Deigan, thank you very much for such a compelling presentation and for putting your stake in the ground, saying quite plainly that NGS is the new gold standard for clinical molecular testing. Let me remind everyone participating once again that you can feel free to type in your comments and questions, and we will deal with all that we can at the conclusion of our program. Which brings me to our final speaker. You'll hear now from Dr. Chris Corless. He's an MD and PhD. He's the Executive Director and the Chief Medical Officer of the Knight Diagnostic Laboratories at Oregon Health Sciences University, where he's also Professor of Pathology. It's been our pleasure to have used Dr. Corless frequently as a source in CAP Today, and I do remember a wonderful cover photo as well with him on that. So with that in mind, I give you Dr. Chris Corless.
Thank you very much, Bob, and my thanks to CAP Today and Horizon for the opportunity to participate in this terrific forum. So before I go any further, I do have some disclosures. I'd like you to note that I have received travel support from Ion Torrent Thermo Fisher. These other disclosures are really not that relevant to today's talk. So I'm going to be discussing with you more on the somatic or cancer side of the NGS story and try to complement Dr. Deigan's excellent presentation. I'll be focusing on our panel-based sequencing approach to cancers and discuss with you our analytical validation approach as well as particular issues that come up with regard to insertions and deletions. I'll also be touching on the very important issue of specimen quality going into the sequencing. And then I want to touch on some other things that we can do to squeeze more information out of next-gen sequencing in the case of cancers, such as looking at copy number alterations and looking for gene fusions by RNA sequencing. Now, what's motivated us to get into next-generation sequencing, and our work in this area goes back to 2012, is the fact that we've got a growing number of targeted therapeutics in cancer, and many of the ones I've shown you on this slide are already FDA approved, but by some counts, there are over 500 compounds in development. And so we have been trying to come up with panels that will cover at least part of this waterfront and provide information about mutations that might indicate that a particular tumor would be sensitive to one or more of these kinds of drugs. And this has really changed my thinking as a pathologist. You know, I used to look down the microscope and see images like these and recognize them for the standard histological diagnoses like lung adenocarcinoma. But nowadays, I'm actually more interested in what are the underlying driver mutations that we might be able to target to benefit the patient, particularly in the setting of metastatic disease. And this isn't true just for lung adenocarcinoma, but also for squamous cell carcinoma of the lung, breast carcinomas, and all of these other malignancies I've illustrated here. And basically across all solid tumors and leukemias and so forth these days, we have a growing list of molecular subtypes that have been recognized and are important to find in order to solicit the best treatment for the patient. We have a number of next-gen sequencing tests that we've brought online. All of these are custom. They're all based on an amplicon type of library, and they're all designed to work with material from formalin-fixed paraffin-embedded tissue. You can see that these are relatively small panels ranging from 23 to 76 genes, and the reason for this is because we've chosen to just focus on those genes that we think are truly actionable with drugs today. But of course, as more drugs become available and more targets are recognized, we'll have to be expanding these panels in the future. So along the way, we've adopted kind of a standard approach for each new panel that we bring online, and I think this is applicable not just for custom panels, but for anybody who's developing or adopting a commercially available panel. We first run 10 to 20 samples of normal tissue DNA, usually from FFPE tissue. And the reason for doing this is because it helps establish what is the background with the panel. There are going to be some false positives that turn up, and we want to recognize those and subtract them out from future runs. We next run between 40 and 50 samples that have known SNVs or mutations, including insertions and deletions, and or copy number alterations. Now, to do this, we collect together a variety of types of substrates. Sometimes we use control cell lines or samples available from commercial resources. But most of the time, we reach into the freezer and we pull out samples that have been previously examined and shown to have a mutation by another technology, for example, Sanger sequencing or mass array sequenome technology. But increasingly, we're also starting to cross-validate from one panel to another using samples that we've found to have a low mutant allele frequency. We could not necessarily confirm it by Sanger sequencing, but when we run it on a separate panel, we can see that the mutation is likely to be real. And in that sense, I'm picking up on the same theme that Dr. Deigan put forward, which is to say that NGS can serve as a gold standard to itself. We also run dilutions, of course, to determine the limit of detection for our panels, and then we, of course, do reproducibility runs as is the standard of practice. So there are some things to keep in mind when you're assessing a new panel. One is that you need to look at areas of low coverage, and there will be areas that are not well covered for certain regions of genes, particularly GC-rich regions, particularly exon 1 of genes. These can be poorly covered by a panel. And it's important to make sure that you don't have any known mutation hotspots in these areas. And if you do, you have to find a way to cross-cover those. What is the lower limit of detection, and how does DNA deamination impact this? So we, along with many other laboratories, have discovered the uncomfortable truth that the longer a paraffin block sits in storage, the more spontaneous deamination occurs in the DNA, causing aberrant C to T transitions. And so in some samples that are older than, say, 3 or 4 or 5 years old, we tend to see increasing background that comes up, and that means we have to adjust our limit of detection for that sample. There are, of course, some samples that are so badly degraded that we can't use them at all, and we have to basically sign it out as insufficient. We also want to keep in mind what is the size range of insertions and deletions that can be detected. The fact is that the software today has improved greatly over the last 3 years, but still there are problems in trying to identify some of the longer insertions and deletions. In the case of our lung cancer assay, we're concerned about missing critical deletions that occur in EGFR exon 19. So we run a backup assay, a high-resolution melting curve assay, that allows us to pick these things up and make sure that we haven't missed it by the NGS approach. Now, the other really critical thing about doing next-gen sequencing on tumor samples is assessing the material that you start with. Now, here's an example of metastatic melanoma in the lung. It's an obvious nodule. It's very easy for the technician to scrape this area that's been marked on the glass slide, and we're going to have material that is essentially 90% tumor cells and no problem identifying any mutations that are present. On the other hand, we not uncommonly receive samples like this, where we have colon cancer metastatic in the mesentery. There are just small clusters of tumor cells here and about, lots of inflammatory cells and stroma to interfere. Even if we concentrate on just this region of the slide, we're going to be pushing up against the limit of the assay. And in our lab, we define the lower limit of an acceptable sample as having 20% tumor content. Below this, we really start to get uncomfortable because we have to keep in mind that this is an eyeball guess and not an exact count of of tumor fraction. And so there is the possibility that we sometimes underestimate or overestimate the amount of tumor present. Some samples come through and they're simply inadequate. This is a transbronchial biopsy. 3 of the fragments are present, but 2 of them have no tumor in them. The left corner has a little focus of tumor in the smallest fragment. But frankly, this is not enough material to work with, and we will turn it down up front rather than try to attempt sequencing it. Now, one of the things I think is kind of fun is to try to compare the what we estimated to be the tumor content on the microscope versus what we actually get out on the sequencing. So here's a corner of metastatic colon cancer in a slice of liver, and we've dissected out the region that's been circled. I estimated 60% tumor content on the microscope. What we found by sequencing were 2 mutations, NRAS and P53, that were in the range of 26 to 27% mutant allele. Now, if we assume for the moment that both of these are heterozygous mutations, then we could back calculate that the actual tumor fraction in the sample was between 52 and 54%, which makes me actually quite happy because it's fairly close to the 60% that I estimated. Now, it doesn't always work out so nicely, I have to admit, and I will say that as a general rule, and it applies to me as well, pathologists tend to overestimate how much tumor they have on the starting material. And this is something we all need to keep in mind when doing somatic testing. Now, one of the other things that we've worked on in the past year is looking at copy number alterations. And we developed our own algorithm, which we published in Journal of Molecular Diagnostics in January. And what we've done here is to assess the number of reads that we're getting back for each amplicon. So each little colored dot here on the slide represents one amplicon. And we're looking at the number of reads we get back for each gene across 25 genes in a panel. And most of them are lining up right around 2 copies in line with the arrow on the left. But 2 of the genes have amplicons that are yielding a lot more reads, FGFR1 and HER2. And these correspond with gene amplifications. There was a known HER2 amplification in this tumor, and we were able to confirm the other one as well by FISH. So by this kind of approach, we compared our results with the algorithm versus samples that we had a known FISH status for 1 or more genes, as well as samples that had been run through a microarray. And we were able to come up with what we feel is a reasonable algorithm for doing copy number alteration assessments. But we also were interested in looking at what was the lower limit of detection with this approach. And here's a sample that we took that had 80% tumor nuclei and a known HER2 amplification. And we diluted it out with its normal matched DNA and sequenced it to the tune of 64% tumor, 32% tumor, and so on. And what we found was that in the sample, we could get all the way down to 16% tumor and still see that HER2 amplification. Well, that's comforting. But when you look at another gene on the other side of the slide, DDR2, it was also overrepresented in the tumor, but not to the same extent. So in that case, in order to see amplification of that gene, we need 30% tumor. And then looking at RB1 in the middle, we had loss of an RB1 allele, but we needed 56% tumor content to be able to detect that. So through this approach and some additional statistical analyses that we've done, we've determined that if we have 60% tumor, then we have an excellent chance of picking up 1 copy loss. And if we have 30% tumor, we have a good chance of picking up anywhere up to 2 copies that have been lost. But below that, we're really treading into territory where we may miss a significant copy number alteration. And so this is important to keep in mind. Now, here's a sample that actually had very poor quality DNA. I actually would be hesitating to sign this case out, except for the fact that even despite the poor sequencing, we had EGFR jump out sky high in this particular breast carcinoma, which is a bit of a surprise. So we went back and we confirmed by FISH, as you can see with these pink signals, that this tumor indeed had EGFR amplification, and it was HER2 negative. It turns out that a small subset of breast carcinomas have EGFR amplification, and there's now an ongoing clinical trial to see if targeting that particular marker might be helpful to these patients. Now, lung cancer, in many ways, is becoming the focal point for next-gen sequencing in the area of tumor biology, and that's because we've got so many different targets that have been identified in recent years. There are some that, for example, already have FDA-approved drugs, the ones that are highlighted in blue, EGFR, ALK, and ROS1. There are many other targets for which we have FDA-approved drugs, but they've only been approved in other cancers, not yet in lung. Nevertheless, we think that these are good targets. There's already early evidence to support that, and so we're interested in identifying these. And finally, we have a number of targets for which there's drugs that are still in clinical trials. So what we try to do with our panel then is to confirm or identify mutations across as many of these genes as we can, but we need to also keep in mind that a number of the targets are also involved with gene fusions. These include ALK, ROS1, RET, and the NTRK and FGFR genes. So this raises an important question, and that is, can we adopt next-gen sequencing to pick up these gene fusion events? Now, what are the other alternatives? Well, we could potentially use immunohistochemistry. It has been shown, for example, that if you're looking for ALK fusions or ROS1 fusions, antibody staining will highlight those tumors that overexpress these markers on the basis of the fusion. But that approach is not going to work for many of the other genes of interest because they're already expressed at significant levels in many tumors and in normal tissues. So that's not really going to do it. Now, FISH, of course, is still the gold standard for looking for structural alterations, and we could use that. But if we want to cover the complete waterfront today for all non-small cell lung cancers, look how many different FISH assays we have to run. Frankly, we're going to run out of tissue, and we're going to run out of money before we can do all these FISH assays. This just really isn't that practical. So instead, we've developed an RNA sequencing panel, and this is work conducted over the last year in collaboration with our colleagues at the University of Pittsburgh, Dr. Marina Nikiforova and her colleagues, as well as some folks at Ambion Thermo Fisher. And the approach here is outlined. First, we start to extract RNA from the formalin-fixed paraffin-embedded tissue. We treat with DNase, and then convert the RNA into cDNA. And from there, we have primers that are multiplexed into a pool that will, we hope, amplify fusion genes that are present and allow us to pick them up by next-gen sequencing and confirm by looking at the integrative genome viewer that the sequence actually spans the breakpoint that we expect. So here's the panel, and on the left side are the target genes, and on the right side, on the right column, are all of the partner genes that we're attempting to try to find to these targets. We have down below a number of these targets also being assessed for their general expression, and of course we have control genes to make sure that the RNA that we started work with is working well. But the question comes up for a panel like this: how do you validate it? And what we did is we raided our freezer, and we were able to work through the freezers at the University of Pittsburgh with our colleagues there, and we came up with as many different fusion events as we could to try to validate this panel. And so all the colored pairs here are ones that we've been able to show actually work. Does that mean that the whole panel is validated? No, not in my mind. It means that the ones we've identified and shown that they can work, those are validated, but the rest, at this point, are largely theoretical. Should we not use a panel like this because it's not fully validated? Well, in my mind, this is still the best, most cost-effective approach for looking for these kinds of fusion events that we have right now. I would say that there's a nice commercial opportunity here for companies that are interested in developing materials for validation, because we've got lots and lots of these fusion events that I think everybody would be interested in identifying in the future. And so until we have those, we cannot regard a panel like this as being 100% validated. But we are using it, and we have had positive impact. So, for example, an FGFR2 BIC1 fusion turned up in a cholangiocarcinoma after we started using this panel clinically, and in a sense, that added to the validation. We were able to show that that was present by Sanger sequencing after we found this. Now, the final comments I want to make are that for anybody who's venturing into next-gen sequencing, it's important to keep in mind that you're going to have a very robust genomics database of one sort or another. You might start with something that's commercially available and adapt it to your own laboratory, or you might invest in developing one of your own. We've put considerable resources into our clinical genomics database over the last few years, and it's an ongoing project. Frankly, it's quite expensive. Now, we do rely on public resources to help us determine which mutations might be important and what their sensitivities might be to various drugs that are out there. I'd point you to mycancergenome.com, which is one resource. MD Anderson also has a site that's very nice and allows you to look up various mutations and read about them and their potential drug sensitivities. And finally, there's also a very nice site that's been put together by Washington University St. Louis called Civic. I refer to all of these sites, but one concern I have is that they have the common mutations, but they don't necessarily have the rare ones. And so for that reason, we've also been backfilling our own database with information about rare variants, and that helps me when I'm doing my sign-out. So, for example, here is an example of a duodenal adenocarcinoma metastatic to the liver. We found a rare codon 146 of KRAS that was mutated. The database goes ahead and prompts me when I'm reviewing this case and tells me that this mutation is correlated with resistance to cetuximab and panitumumab, but may be sensitive on the basis of preclinical data to some other inhibitors. And so this is helpful if you're trying to streamline your sign-out to have as much information on hand as you possibly can. Now, that being said, there are, of course, going to be unknown variants that pop up in cases, and this happens to us every week. This is one case where there was an unknown variant in the KIT gene that was found in a thymic carcinoma. It had never been published. We'd never seen it. No one else has seen it. But it was in the kinase domain, and it was 4 codons away from a known hotspot, and it's a non-conservative substitution. So, because the patient had no other treatment options, we suggested that the patient get treated with a KIT inhibitor. And as you can see on the right side, the PET scan already began to show a marked diminution in signal of the tumor at 12 days of treatment. And I'm happy to report that the patient continues to do very well 15 months later. So just to summarize then, analytical validation of cancer panels should, in my view, include evaluation of normal DNA samples, looking at samples with a wide range of mutations, particularly large insertions and deletions to challenge the panel, as well as looking at samples with low mutant allele frequency. It is important to correlate the sequencing results with the tumor input. If you have 90% tumor and you're about to report out a mutation with 3% mutant allele frequency, you might want to reconsider. Copy number alterations can be detected, but there are limitations to this. And again, you have to correlate with your starting material. And finally, RNA sequencing could be useful for detecting gene fusion events. But again, there are limitations there, particularly the challenge of trying to validate approaches like this. So with that, I'm happy to turn it back over to Bob and look forward to questions.
Dr. Corless, thank you very, very much. I think our presentations today have yielded as many data points as an exercise in next-gen sequencing itself, and so it's now my privilege and my challenge to try to present some of the questions and comments to you in some of the time we have remaining. We had slated this for 1 hour, but thanks to our vendor, we can continue. And I would like to spend a few minutes with all of you going over some of these questions and comments. Again, I want to remind you we will post this webcast in its entirety on captodayonline.com in about a week to 10 days. And if you don't get a question answered, we'll be happy to entertain them and do our best through our normal magazine Q&A mechanism and other mechanisms to get you the answers that you want. So while I let Dr. Deigan and Dr. Corless catch their breath, I want to start with one question for Dr. Frampton, and this is, I think, a fairly easy one. Does Horizon have a U.S. distributor, Dr. Frampton?
Thank you, Bob, and thank you for the question, and also, I guess I should also thank both Dr. Deigan and Dr. Corless for their fantastic talks. We sell direct into the US, and so you can come. What's nice is the website's right up there. You can go straight to the website, and you can get anything you need straight from the website. I think it was. If I can just add a little bit of words from my side of things, it was fantastic to get both the perspective of both Dr. Deigan and Dr. Corless, and this is really what I believe. As a community, NGS has arrived so fast, and it is a steam engine in being adopted by labs. And together, it's critical that we share experiences of how it is being adopted into labs, and also for us as a reference standards provider, please do interact with us either through the Q&A, which has got lots of questions coming up, or come to the website. We've got scientists on our live chat, and because we are making RNA standards, we're making standards for cell-free DNA. We've got the genome in a bottle standards. We're looking to work as closely with the community as we can to provide standards that are required in the field.
Dr. Frampton, thank you very much. So indeed, in addition to the NIST sample in a bottle, Horizon has a great deal of material and also a great deal of science behind their offering. So I would encourage you to go to their website. There are also very many of our scientific meetings today in the U.S. and elsewhere, and you can easily find someone to have dialogue with. Dr. Deigan, there is a lot of interest in understanding this genome in a bottle from NIST. It's pointed out to me that there's a plan to actually publish the sequencing data from this genome in a bottle for free online. But tell us a little more. This is a genome, this is one single sample that would serve for any purpose. Tell us a little bit more, as you understand it, what NIST will be providing.
Sure. So I would of course defer everybody for more specific questions to the actual Genome in a Bottle website and the individuals involved there. I will say that I have not been directly involved in the characterization of the Genome in a Bottle samples. My understanding is that what will be released very soon is a single sample, which they have been working on for quite a while. And hopefully what will be utilized fairly quickly from various clinical laboratories will be consensus single nucleotide variant, and I believe small insertion deletion calls. And I know they're working on other types of structural rearrangements, as well as working on the characterization of additional samples. And I believe that there are certainly at least one or multiple trios of samples that they are hoping to release as well. I know that every time I've seen the updates and heard about this project, I've gotten very excited and am definitely interested in getting our hands on some of these samples when they're available and comparing our sequencing pipeline with some others. So that's basically the extent of my knowledge. And like I said, I would certainly defer any interested parties to the website and the relevant individuals who are directly involved.
Yes, I think that's a very good answer. And it's my understanding that this is part of a CDC project. They're working in concert with NIST to get some of this information out there easily and publicly. Let me turn to a question that I think is particularly a good one, and it's been asked with several variations by several of the participants. And it fundamentally, I think, gets to a core question. As people are reviewing the literature for some certain single mutations of genes of interest, particularly from formalin-fixed paraffin-embedded tissues, we're finding that when you really do compare different kits and methods, we do not find one study which shows 100% concordance, even when the study uses Sanger sequencing as a gold standard. I think in many ways that's exactly what both of you were speaking about today. But Dr. Deigan, tell me about the correlation from different methods.
Yeah, so that's something that we certainly see all the time, and it is important to keep in mind whether you're comparing, as I presented, two different next-gen methods, and certainly Dr. Corless alluded to that as well, or two different methods between each other, that you can very easily have both methods working perfectly to the extent of their capabilities, and yet one provides a positive result at a given variant position and the other provides a negative result. And so I think just knowing the method that you're using and understanding the assay in its entirety is very much the purpose of validating a test. So I'd encourage laboratories certainly not to shortchange themselves with the upfront validation, but just know that simply differences in methods can lead to those types of what some may get unfortunately concerned about as being discrepant results, and yet in their own independent setting, they're both perfectly acceptable for the methods that are being used. So I would just caution everybody to really know what test they're doing and understand how it works.
Chris, could I ask you to give some comment to that particular question?
Sure. I mean, we can start with a basic fact, which is there's no such thing as a perfect test. So once we recognize that, then we can say, all right, we expect that there are going to be occasional differences. But for the most part, I have been impressed. I mean, again, we've compared against Sanger sequencing and mass array and occasional other assays that we've gotten. And time and again, our next-gen panels have really stood up and shown us those same mutations with very, very few exceptions. Now, we're not doing the whole exome. We're just doing small panels. But still, the reproducibility across these methods has been quite strikingly good. And I actually take heart in that rather than focus so much on the rare exceptions.
Okay. We've had several questions around the quality score of 500 from you, Josh. How did you decide on that as a threshold for the variant call, as opposed to a number that was lower or higher, 400 or 600?
So yeah, we spent quite a lot of time, and like I mentioned, looking at really a year and a half's worth of data in comparison with Sanger sequencing in order to derive that cutoff, and certainly have actually set that in a conservative fashion. And so what we've actually seen since then, because we really have only been confirming variants with a quality score of 500 or below, is it really, I think, maybe more so above 200 or 300, we've still been able to get complete concordance, and it's only when you drop into that very low level that we've noticed the occasional discrepant result. And so, even saying that, I think the quality score by itself is not the only thing that we look at. We certainly compare it with coverage. We look at where the variant might be in relevance to the rest of the gene, where a particular variant may be, and whether it's been seen in other samples in the run. But it has been a fairly useful metric for us for the last couple of years. And I will just say that we set that a number of years ago, but to date, even though we've accumulated some additional data, haven't yet felt comfortable moving that down on a more formal basis.
Good. Thank you very much. And Dr. Corless, for you, of course, as you can imagine, there's lots of questions around your samples, including, for example, can you give us a general rate of sample rejection of fresh frozen paraffin-embedded samples when you look to perform NGS on them?
Sure. Yeah. Actually, it depends very much on the nature of the tumor. So the worst offenders are transbronchial biopsies, because you tend to get a lot of normal tissue and relatively little tumor, and there's not much tissue overall. So that probably has the highest rejection rate, and it probably is about 10 to 15%. We also sometimes have to reject pleural fluids and acidic fluids because they have too many inflammatory cells in them. Again, that's at about a 5 to 10% rejection rate. Across other kinds of cancers and other types of samples, the rejection rate is under 5%.
Very good. Thank you. And I would be doing everyone a disservice if I didn't point out that Dr. David Rim, who I thank for participating with us today, reminds me to remind you that in a study published last year in the Archives of Pathology, the CAP did a study regarding the accuracy of the estimation of percentage of tumor cells, showing that inaccuracy can often exceed 20%. And so that is an article well worth reading, it seems to me.
Yes, that's an important point, and that's why I like to look at the sequencing and look back at the tissue to make sure that the two are really in agreement with each other once you look closely.
Yes, and that particular paper is Verret et al., 2014, Archives of Pathology and LabMed, pages 1545 to 1549. And thank you, David, for pointing that out. At the same time, I wish to point out as well, in the April issue of that same journal, which is, of course, the CAP's journal, there is an extensive article and a wonderful discussion about how the committee that put together the NGS checklist went about putting it together, including discussion and debate and pro and con about the eventual product of that checklist, and I'd recommend that to everyone. Finally, I want to because we have so many clinical questions here, Chris, what do you do with these results vis-a-vis your reporting? Do you try to put everything all in one place? Do you use a lot of additional addendum reports? How do those go together in the reporting product from your laboratory?
Sure, that's a challenge. If you've got NextGen coming out on 1 week, but you've already got immunohistochemistry or FISH results that have come out previously, then it is a challenge. Sometimes the clinicians complain if everything's reported separately. Our system is not ideal. We basically have to report each test separately, otherwise it creates problems internally. But we do try to cross-reference from one report to another so that clinicians are aware that other results are out there. And, of course, the latest report will incorporate all of the information as best we can to come to the final conclusion.
Thank you very much. Are those reports then posted to an EMR in your institution?
Yes. It's all electronic, and we also have PDFs available of the reports.
Very good. Now, let me ask you this. Do you typically validate 40 or 50 positive control specimens for each genetic locus in the panel? Or do you typically look for 40 to 50 positive control specimens for the whole gene panel?
Yeah, it's for the whole gene panel. No,
yes,
we could not try to do every single locus. There's no way.
That's very good. I did want to clarify that, though, for our audience. Dr. Deigan, you had some very interesting examples and of particular cases and particular facts from your laboratory. Here's one question. Were you able to figure out why Sanger sequencing did not pick up the homozygous mutation that you saw by NGS? Did you, for example, try using alternate PCR primers for that Sanger sequencing?
So yes, actually, in that scenario, and then really the other scenarios, the Sanger sequencing results originated from outside reference laboratories, and only the exome sequencing was performed at UCLA. So when we communicated back with the ordering clinician, who then I'm sure had an interesting conversation with the outside reference laboratory, I know on at least one or two of those situations they went back and looked at the traces again, or resequenced the gene, and I can't remember the actual outcome and the rationale as to why they missed it. I believe one may have been a sample mix-up, but all of those possibilities could exist. But yes, none of that Sanger sequencing was done here in our lab. Okay. We've had several questions about the provocative statement about NGS becoming the new gold standard. And I'm going to combine several of them into really one, which is, given the fact that we do have different platforms, although I must say we have many fewer platforms than we have in most of the other modalities of pathology.
But given the fact that we do have more than one platform, and they show a lot of lack of concordance between methods and platforms, what will we really be gaining by trying to establish NGS as a new gold standard? I'll ask both of you to respond to that, if you will.
Sure. Would you like to start, Chris, or should I?
I was just going to suggest you do that.
That's the sign of a very good question, isn't it, gentlemen?
It is. Right. It is a very good question. Okay, so I've been following in the literature papers that have come out in the last 2 to 3 years that have compared the platforms that are out there, and I've been pleased to see that with each paper there's been improvement, better agreement, better performance characteristics, and I think that they're much more robust now than they were when they were first being brought into the market. And so I have confidence in the next-gen sequencing from the major vendors right now, and I actually don't lose a lot of sleep about the idea that one is very different from the other. I think that they're all working quite nicely, and frankly, I prefer all of them to Sanger sequencing because I think Sanger sequencing has a lot of its own issues.
Thank you. Josh.
Yes, and I would certainly second everything that Dr. Corless just mentioned. You know, and just when it comes to next-generation sequencing, certainly a method can be a gold standard method and have sources of variability or known complications that it involves when you utilize it in the clinical laboratory. And it's just important, and I'll say this, you know, again, to understand what those are. When I think about both in the germline and somatic context, all of the situations where one might want to compare a given method to another, I think one is more likely to have a result that is found by next-gen sequencing if the lab understands and kind of modifies their pipeline to reduce false positives, for example, that is found by next-gen and then may not be picked up by another method. And so part of that is just why I would argue that it is really kind of the method that I think everything should be compared to, simply because I think if utilized appropriately, its window of visibility, I think, is much greater into the actual genetic variation than any of the other methods that we have in our toolkit.
Thank you very much. As I asked that question and as you answered it, it reminded me historically that the very first effort in proficiency testing done by the College of American Pathologists in the late 40s was some of the very simplest assays that were available, and the results were so discordant that they didn't even publish them. They were so embarrassed to see how there was no agreement from even the finest labs in the United States. So I would say on a relative basis and a historic basis that next-gen sequencing is doing quite well. Let me finally come with a couple of final minor points here. I know it's getting a little late, but we've got a lot of people hanging on here who'd like to hear these questions. First, for Dr. Corless, do you ever use digital PCR to validate low-abundance mutant allele frequencies?
No, we have not done that. It would be a very good way to approach that issue, but we have not employed digital PCR.
Can I jump in and comment?
Yes, please, Jonathan.
So that's one. It's one approach we've taken for our reference materials. And so whilst you can do a number of validations on the well-characterized genomes with next-generation sequencing, we're also taking it that step further to actually validate the hotspots, so like the KRASs, the NRASs, using digital PCR, because it gives you an orthogonal method. So if we are developing a standard that's down at 1% KRAS G12D, we can show that with digital PCR, and it's a nice way to then, and you can then look at that again and compare to, say, a MySeq or an Ion Torrent. So I think it may not be kind of feasible to do at the sort of the clinical level, because having a digital PCR set up as well as having an NGS set up is always going to be a challenge in a lab. But perhaps it should be pushed onto these well-characterized reference materials.
Thank you. Now we have another question right from the field, which asks the following of both of you. What about checking that each amplicon has sufficient coverage as part of the QC for each run? We find that if the sample performs poorly, then there will be amplicon dropout. So we have the number of reads for each amplicon checked as part of our analysis pipeline. Does that sound like a prudent thing to do?
Yeah, we do that same thing. We monitor what is the overall level and have an automated part of the report that alerts us to all regions that have less than 100 read coverage.
Very good. Thank you. Chris, do you do something similar?
Oh, that was Chris here.
I'm sorry. Yes, Chris, I do. Josh, do you do something similar in terms of the coverage of the amplicon?
So because we're not using amplicon specifically, but using captured regions from the entirety of the exome, we do look at things like average coverage across the exome, coverage across all relevant disease gene regions as one of our quality control metrics that I hadn't shown before. Certainly with an exome sequencing capture kit, there are regions that are not adequately sequenced. And so one of the things that we provide to any interested ordering clinicians is an average gene coverage on our website so that clinicians who are interested in ordering exome sequencing can just go there, look and see if their gene of clinical interest is covered well or not covered well, and then make test ordering decisions based on that. And thankfully, due to improvements, there are quite a number of genes that have gotten a lot closer to 100% or are now 100% sequenced using exome capture.
Very good. Thank you. And while I have you, Josh, on the phone, I've got a question about how you do deletion and duplication studies in conjunction with your panel NGS when you're studying multiple targets.
So for us, we actually don't currently offer germline panels. Really, we only offer exome sequencing on the germline setting. And we don't currently report any deletions or duplications much beyond about 10 base pairs in size. We do also have a couple of somatic panels, but also mainly focus on these small variants.
Okay. And finally, I'm going to ask one final question and then ask for comment from any of our speakers. There's a lot of interest, of course, in how you're enriching your targets of interest, and as well as other technologies you're deploying to enhance your assays. So let's start first with Chris. Are you using some enrichment technologies for your targets of interest?
No, we're just doing standard DNA purification, or in one case RNA purification, and then amplicon-based library prep. So there's nothing beyond that. Nothing beyond that.
And Josh, for you?
Yeah, same. We use a commercially available exome capture kit to certainly select away all the non-coding regions that we're not interested in.
Okay. And Dr. Frampton, in your experience, these tend to be then sufficient steps to get accurate results. Is that true?
Yeah, I think that's definitely true. I guess when the enrichment steps will start to come in play, and it will probably be over the next 12 months or 18 months, is when NGS wants to get into the cell-free DNA space. So then you are going to have to start doing enrichments on the targets that you're looking for, because you can have such low copy numbers to actually target. I don't know if that's a comment that Chris or Josh want to comment on before we sort of wrap up. But I think that's where enrichment will become critical, is if you want to use the NGS for detecting and circulating free DNA.
Yeah, I think that's the case, and it's also true for CTCs, circulating tumor cells.
Right. Well, we know that free DNA dominated the headlines coming out of the AACR meeting. Is that something that I'm assuming you must have at least some research projects and interest in both of your laboratories at UCLA and at Oregon, yes?
Definitely here at OHSU, yes.
Yeah, okay. Here we're interested in that as well.
Okay, very good. And with that, I think in deference to your schedules, I'm going to wrap this up. We've had, as you can tell, a very, very rich day, both within the presentations and in this Q&A. I want to assure you we plan to have the Q&A recording also online for you all. So to sum up, I want to thank Jonathan Frampton and Horizon Diagnostics, Dr. Josh Deigan from UCLA, and Dr. Chris Corless from Oregon Health Sciences for giving us a wonderful CAP Today webcast today. You should know that we will, as I've mentioned many times, post this webcast in its entirety at captodayonline.com. You may receive a brief online survey about the quality of this webcast after it's over. I'd encourage you to fill that out. We are still relatively new at this game, so any suggestion or comment that you make we'll take to heart. And once again, I want to thank our speakers, Horizon Diagnostics, and thank all of you for participating with us today. Good day.